All right, so here in chapter 3, point 1 and part 2, we're going to be looking at bacteria and archaea and looking at their structures, what they have that are similar, what they have that are different. And of course, we're going to be focusing primarily on bacteria because bacteria are what are pathogenic and what can make us sick, and archaea don't make us sick. So remember, bacteria and archaea are prokaryotes. Remember, pro meaning before, carry, kernel, or the nucleus. So before the nucleus. So they do not have a true nucleus. They may have a nucleoid region, but they do not have a membrane-bound enclosed nucleus like eukaryotes do. Eukaryotes, remember, those are true nucleus. Bacteria and archaea have DNA or RNA that is inside the cell. However, they do not have that defined nucleus. Eukaryotes will wrap the DNA around a histone protein, and bacteria do not. And so the way that works is, let's see, I've got to bundle up my chromosomes because they're very, very long so that I can store them, right? So in a eukaryote, they're going to wrap themselves around these little dots here, which are kind of like histone proteins. And you can kind of think of that like being, if you have the little plastic thing that you wrap your Christmas lights up on, right? You coil them around it so that you can store them better, all right? So that's what histone proteins are. And so eukaryotes have those to coil their DNA around. Prokaryotes do not, all right? They're also going to have wall structures or cell wall structures that are different than eukaryotes, all right? Not all eukaryotes have cell walls. Humans don't have cell walls, right? So they're going to have cell wall structures that are different. Bacteria have cell walls that are made of peptidoglycan. You're going to find out that's really, really important and helpful for us. It's helpful for the bacteria too, but it's helpful for us in identification and in treatment. Archaea walls, and peptidoglycan makes it strong. Archaea walls are also strong and tough. They're just made of other particular chemicals there, all right? Bacteria do not have complex membrane-bound organelles like eukaryotes do. They have some internal membranes, but they're not membrane-bound surrounding membranes surrounding those organelles. All right, and so in a eukaryote, those organelles all have a specific function. In a prokaryote, metabolism things are just kind of done in general. Like I said, we're going to focus on the features more of the bacteria because that's what we're going to encounter in the clinical environment. So we're going to briefly touch on the differences for archaea. All right. All bacterial cells possess a cytoplasmic membrane, cytoplasm, ribosome, cytoskeleton, and one or several chromosomes. Okay. The majority have a cell wall and a glycocalyx, and we'll get into a little more detail about what these things are. There are some features that bacteria can have, but not all of them have them. These are going to be things like fimbriae, pili, flagella, an outer extramembrane, plasmids, inclusions, endospores. There's some new things that we're learning about called nanowires and nanotubules, and they can also have some microcompartments in them. So this is a really good generalized picture of a bacterium, what a general bacteria will have. Okay, so this goes from the cell surface that's going to have some things to help adherence for adhesion to other cells or other surfaces. We're going to have the fimbriae. Oh, here we go. This fimbriae here. There's going to be helping it to adhere. Okay, we're going to have other appendages like this flagella, which helps with movement. Okay, provide motility. And then on another side, we may have a pilus, okay, which looks kind of like a drinking straw. This is a pilus right here. All right, that's used in order to transfer DNA and things like resistance to antibiotics. All right, so that's something important for us. From the outside moving in, you're going to see you have a capsule layer, a cell wall, cell membrane, and then a water-based environment here. Okay, that fills kind of the entire cell, and we'll cover these things in more detail later on. Within the cytoplasm, we've got ribosomes that are the sites for protein synthesis, all these little fine spherical specks here. This squiggly spaghetti looking stuff is the bacterial chromosome, okay? And so we say that's the nucleoid region, all right? It looks kind of like a mass of spaghetti noodles there. It's going to have an inclusion or granule sometimes in there, which is a large sphere where nutrients can be stored for later use. There can also be a small circular plasmid DNA, which is going to be down here. Okay, it's a loop of extra, so to speak, double-stranded DNA that contains some extra genes. And so you can kind of look at this picture and see all these other things. There are things that we will talk about that aren't shown, particularly one that's not shown here that we're going to talk about is going to be the endospore. Okay, the endospore, that's going to be important. All right, so let's talk about some bacterial shapes and arrangements. Most bacteria are going to be unicellular, uni meaning one, so they're single-celled. Each individual bacteria cell can carry out all of the necessary life functions. So each cell can carry out reproduction, metabolism, or obtaining nutrients. So this is very different than a multicellular organism like we are, okay? Because we have special cells to do all of those different things, okay? So most bacteria here, we said, are independent, single-celled, and unicellular. They can act together as a group as a biofilm, which is really a kind of interesting, neat thing. I put a link to some information on biofilms. If you really find them very interesting, go to the TED Talk and look at the biofilm on quorum sensing. You can just Google that. I'm not linking it because it's not required, but it's really quite interesting how it's almost like a little commune, and these bacteria live and function together as one, and it's protective for them, and then they communicate in these really weird ways, okay? They can also communicate through some nanotubules. The size of an average bacterium is about 1 micrometer. That's a micron. Micro is 1 × 10⁻⁶. Now that's kind of hard to wrap your head around what size that really is. And so there is a link that you need to go, and it's not really a video, but you kind of slide through it. It's called cell scale and size, something like that. And you'll click on that link. It's in the little extra folder of chapter animations or something like that, I called it. Be sure to go to that one. That's actually not from the textbook, but it's really good because you can kind of compare all these different things we're talking about with other things like a grain of rice. And it really helps you to wrap your brain around the sizes that we're talking about. All right, so average cocci. Cocci is going to be berry-shaped or round bacterium, 1 micrometer. A rod, which is going to be what we call a bacillus. All right, you're going to be learning all these terms, which is going to be about twice that size. But they can also, and there's actually going to be 3 to 5 basic size, cocci, bacillus, and then we'll look at spiral. Okay, and then we'll look at groupings. But there are some other shapes and sizes, but there are some that actually have variable size and shapes of the cells within a species, and that's called pleomorphism, which is really kind of neat that it's kind of like us, they don't all look the same, okay? Can make it harder to identify though. All right, so here are these 6 basic shapes, okay? You are going to be expected to know these shapes in lab and in lecture, and to know them by their scientific name and what they mean, because it is very, very, very helpful in the lab, because the name of the bacteria often tells us what the shape is, what color it may be expressing, and it really helps us to be able to identify and classify them. Well, why do we want to be able to identify and classify? Why is that important? Well, we can't treat if we don't know what it is, right? So we got to know what we are infected with first, then we can treat it correctly, okay? All right, so here we have cocci or coccus, okay? And so that's a spherical, and I always say ish because they're never perfect. Now, some of them are perfect. These right here, these are really nice little round purple circles, okay? This one happens to be staphylococcus, all right? Staph, by the way, is cluster-like, and we'll get into that again, but it's like a little cluster of grapes that you can see here, okay? So staph part, meaning the cluster, and cocci or coccus, meaning circle, all right? So it tells you what you're looking for. So if I give you a bacteria and I tell you this is a staphylococcus, you know you should be looking for, in the sample, you should be looking for clusters of little round circles, okay? Then we can even add to that, this is gram-positive, okay? So you would know to look for something that's purple, all right? So it's really, really very helpful, and so it's very, very important for you to know this terminology, okay? Okay, so moving on from that. They can be little perfect spheres, but not always. All right, so after that we have bacilli, okay? And the typical bacilli shape is going to be more like this right here, which is in a strip formation, which means chain. This is your classic bacilli here, okay? But if you'll notice, not all of these are straight. Some are kind of curved, and you'll find in lab that you have to really watch for those kinds of things. And what you'll have to do is kind of look at the predominant form that you see. If you see this one little curved one and looked at that, you might want to say, oh, that's Vibrio, because Vibrio here is curved. But most of them are straight. So then we would want to say this is a bacilli or bacillus, okay? So a cell that is rod-shaped, we call that bacillus. Now you need to be aware that there's also a genus named bacillus with a capital B, lowercase b for the shape. Okay, so if it says bacillus in the genus name, you know it's for sure going to be rod-shaped. Okay, a good example of this is going to be over here. This is Legionella pneumophilia. This is a strain, I believe this one causes Legionnaire's disease. All right, so after bacillus, let's see. We are going to be moving on here to our, well, I was thinking about the coccobacillus. And I don't really see a good example of that on here. So because life is not complicated enough, in science it always has to be a little more complicated, right? And if you've had me before, you've heard me say that, like this is why we have to have 3 names for everything. You can find some that are intermediate between a cocci and a bacillus, okay? And so that would be a coccobacillus, and they're kind of short and fat and stubby, a little more round than a typical bacillus, but a little more long than a typical cocci. So we do have some that fall into that particular category, okay? Sometimes they're even club shaped like this, all right? It just depends. All right, so Vibrio is going to have a curved shape to it, almost like a macaroni noodle, okay? Vibrio, so you're going to have single occurring rods. They're gently curved or slightly curved. Vibrio vulnificus, Vibrio cholera. Those are just some examples there. Next, we will have Spirillum. Okay, Spirillum you can see is kind of a short and wide curve here. Okay, it's a helical shape, rigid, twisted along the axis, or a corkscrew would be a good way to say that. An example of that we have right here, and this is a good example. You can see how it's thicker than, say, the spirochete, okay, which is important. And so this is your Spirillum, and this is Campylobacter jejuni. All right, so that's an example of a Spirillum, okay? And then next we have spirochetes, okay? And these are going to be longer and thinner, all right? And these will often have flagella, Spirillum, and I'll often have the flagella on the end, okay? So spirochetes are just longer and skinnier corkscrews. It's a little bit more flexible, looks a little more like a spring, okay? And then a few bacteria, usually the ones that are going to begin with myo, look a bit like yeast, okay? A yeast will have a filamentous form, and it will have a more rounded type of form, okay? So that's going to be a type of fungi. And so these are yeast-like structures, these filaments, okay? And this happens to be streptomyces, okay? And so when you see that MYCE, or you need to be thinking fungal. Okay, sorry, I had to pull my words together here. All right, and so these are the basic shapes that you're going to need to be familiar with. Be sure you know these. Okay, so that's shape, and now we're looking at arrangements or groupings, and students always mess this up. I've traditionally had this little lab thing where you had to name them and they get the names mixed up, okay? Grouping is a little bit different. Grouping is how the cells remain attached after they're duplicated. Shape describes their morphology, what their actual shape is. Grouping is how they're put together, okay? Be sure you keep that straight. So how they remain attached after dividing or growing. All right, so cocci can be found single, so just single little circles here. It can be found as a diplo where you have 2. It can be found in a chain. I've already said strep means chain, so a streptococci would be a chain of cocci, okay? And then they can also divide along more than one plane, and so then you could have this tetrad. And then we have some unusual arrangements here, the sarcina, and all but the clusters. Here you can have the staff-like cluster, which is this here, which is kind of like a cluster of grapes. So that's what staff means, okay? And then we have micrococci, which is going to be big wads or irregular clusters, okay? So cocci have a lot of different ways they can be arranged. Now, I'm trying to remember if, I guess Bacillus is on the next slide. Okay, so sorry about that. I thought it was on this slide, but I couldn't remember. All right, so arrangements of bacilli. All right, and you can see this one actually has a little club on the end. You will see that sometimes. Do not confuse this with an endospore. Okay, so bacilli or bacillus, singular plural, are rod shaped. Okay, they can occur as a single, a diplo, so two linked together, or strep, meaning a chain. Okay. Palisades is interesting. It's a chain that kind of stays partially attached at the ends and then it folds back on each other by giving a new orientation where they're side by side. And that's what where you're getting this little club shape here from, okay? And you'll see this in bacteria like Corynebacterium. Those will be found in this arrangement. And I will go ahead and take this time to say I would get some flashcards. And all of these bacteria that I'm mentioning, I would make one card for each bacteria. And as we go through the course, put all the information you have about them. Okay, so you could start this one, you could say Corynebacterium, and you could write on the back of it, Palisades arrangement. Okay, and then later on, you might want to write if it's gram-positive or gram-negative. And then you might want to add the disease that it causes, what part of the body it affects, because these are stuff that are going to kind of accumulate along the semester and are going to be really important for the final, all right, and for Lat especially. And if you're going into healthcare field, this is the stuff that you need to retain the most out of lecture is keeping those bacteria that cause what diseases in your mind. So you want to keep these, make your little flashcards and review them all 8 weeks long. All right, so here's another good look at some of these shapes. I really like this picture here. We've got the spirillia and the spirochetes, okay? Spirillia can be found in short chains sometimes. Spirochetes are usually going to be single, all right? And so here we have diplococci. This is a good example, Streptococcus pneumoniae, okay? Now it says strep, so it can be in chains, but this particular grouping would be diplo because there's only two. Okay, and then here's a streptococci, Streptococcus pyogenes, and that is found in a true chain. All right, staphylococci, okay, and so this is Staphylococcus aureus. Remember these names, I think I've already talked about this one. Staph is the cluster, cocci is going to be a circle, and while this is colored green because they're making all the cocci green, aureus means what? Remember? It means gold, okay, and so this would have a yellow goldish color whenever we played it out on some agar. And then here's a tetrad, and this would be Sarcina, okay? There are also some really weird bacteria that can be rectangle shaped, star shaped, square shaped, really strange shapes out there, okay? And so here's our bacilli, and so here's a streptobacilli. This would be Bacillus anthracis, which is the bacterium that causes anthrax, okay? And then here's a flagellate rod, which means it's a bacilli. This is Salmonella typhi, and you can see it has multiple flagella here, and we're going to look at the names of the flagella. And then here's another one down here. This is Clostridium botulinum. This is the bacteria that causes botulism, all right? And then here's our spirals are curved. This is our Vibrio. You can see it's only a slight curve. And so this is Vibrio cholerae. This is the bacteria that causes cholera. Spirillia here. Okay, so this is a true spiral. And this is H. pylori or Helicobacter pylori. This is what can cause a peptic or stomach ulcer. And then down here in the very bottom right-hand corner, we have the spirochetes, okay, which are really more just they look kind of like ramen noodles. Their shape to me looks like ramen noodles. So bacteria have different types of accessory structures or appendages, and these are some common ones. Not all bacteria have them, but these are pretty common. We can divide these into two major groups, okay? Those that provide motility. Motility basically means mobility, okay? And that's going to be flagella or an axial filament, okay? And those that function in attachment or helping to adhere to another surface or bacteria, which is going to be fimbriae, pili, and nanotubes. Okay, so the flagellum functions in motility, okay, or self-propulsion. So the ability here of of a cell to swim freely, okay, and it's going to have three parts. It's going to have the filament, okay, it's going to have the hook or the sheath in the basal body. The arrangement of these actually allows the hook to rotate 360 degrees. All Spiralia, half of the bacilli, and a few, very rarely, cocci have flagella. Okay. So bacteria can have different arrangements for their flagella. This is another one that I see students kind of gloss over and miss, and it always comes back to bite them. So please pay attention to these and learn what these are, okay? So these can have single or multiple flagella. They can be found on the ends, okay? So if it's on the ends of a rod-shaped cell, then that would be called polar. So think of poles. What does Earth have? It has two poles, a North Pole and a South Pole, okay? So polar means on one or both sides. Okay, they can also have them all the way around the cells. All right, so in a polar arrangement, and these first few ones are polar, flagella are attached at one or both ends, and we have three types of those. We have monotrichous, mono meaning one, and that's going to be this guy here that has it attached to one end. Okay, and then we have lophotrichous, and that means bunch or tuft, and so you can see there's several of them on one particular side, okay, and then amphitrichous. Amphitrichous, the way you can remember this one is if you are ambidextrous, you can use both hands, correct? And ambi and amphi basically mean the same thing, okay? So both ends will have flagella, all right? So that is this guy here. Okay, and so these three, 1A, B, and D here, these are going to be your polar flagella. And then we have mono for one, lopho for tuft of hair, or you can think, I don't know, luscious hair, something helping you remember lopho. And then amphi, both sides, because ambidextrous is on both hands, okay? And then lastly, you can be peritrichous, okay? Peritrichous is where the flagella are kind of dispersed randomly all around. Peri means around, and so all around the cell here, okay? So motility is important because this is one thing that we can use to identify pathogens. Pathogens are those bacteria that make us sick. So flagella are really hard to see in the lab. I mean, sometimes you can kind of see them and you kind of have to roll your microscope lens back and forth to be able to see them. But if you have a semi-solid medium, it's much easier to see because you can see the results of that movement. You may not see the flagella very easily, but you can tell that these are a motile species of bacteria. And so you know if they're motile, then they must have some way of moving. So these are some good examples here, okay? The basal body here in A of the flagella is going to be made up of a rod, okay, and rings that are embedded in the outer membrane, cell wall, and cell membrane. The basal body is going to anchor the hook in there. So the filament then is a helical structure that extends from the hook like a tail. So you can kind of see that in the orange part. In B here, we have Pseudomonas aeruginosa. So that's going to come through a light microscope, and this has had some flagellar staining. So you can see it's got lophotrichous, I believe, although this particular one here almost makes it look like it is peritrichous. This could be peritrichous here, all right? And then you can see this is definitely going to be a lophotrichous, so I'm sure this is peritrichous. And then here's another peritrichous here as well. Okay, this is more like what you would see if you had a rather large specimen that stained well. And we do have some special dyes that work better for things like flagella and allow us to see them better. All right, so how do flagella operate, or what causes bacteria to move? These are simple organisms, and so they don't think, hey, I want to go over there like we do, okay? But what they have is what we call chemotaxis. And so flagellated bacteria can detect and move in response to chemical signals. Chemo meaning chemical, and taxis refers, okay, so think taxi. A taxi moves you across town. Taxis is movement. Okay, and so you can have a positive chemotaxis. That means movement towards a stimulus, which is going to be something favorable. Okay, a favorable chemical stimulus. So maybe it's a nutrient that this particular bacteria needs, all right? Negative chemotaxis is movement away from something that is going to be harmful, okay? So it's going to move away from something that is harmful. Now, how do they move? Well, they have the most interesting moves. They're called runs and tumbles, and there is a link for movement on here. I don't remember if it's labeled chemotaxis or flagella, but you need to be sure and watch that one. It shows you and explains the little runs and tumbles much better than I can explain with pictures or in words. So go watch that. It's like 3 minutes, okay? All right, so run, excuse me, is when the flagella will rotate counterclockwise and they will move in a straight direction. When they tumble, they're going to reverse the direction and they are going to move, flow counterclockwise, right? And that's going to cause the cell to move in a different direction. Okay, so here are those runs and tumbles that I was talking about. So when a polar flagellum here, all right, let me put this. When this polar flagella here rotates counterclockwise, it's going to be this way, the cell is going to be propelled forward, okay? And so then when it, I think that actually should be clockwise and then counterclockwise. So when it changes direction, it's going to stop then and do what we call a tumble. And it's you really just have to watch the video to kind of get this. Now, the peritrichus are the ones that are kind of fun to watch. And so if it's peritrichus, all the flagella are going to kind of sweep back towards one end of the cell and rotate as a single group. So they're going to really be propelled forward. And then when they tumble, they kind of splay everything all out all around them. It's really funny looking, but you just got to watch the video to see it. Again, this is kind of a random series of movements, even though it can be in response to chemical signals. Here is random here with no attractant or repellent. So then it's very random. There's no purposeful movement there, okay? Now, if there is an attractant or repellent, it's going to move towards or away from that a little bit more organized, although you can see this is still just kind of messy here to get to where it's going. It's not going in a direct line path. Periplasmic flagella, or what I always call axial filaments. I've actually don't know of any other text that uses periplasmic flagella, but if there's more than one word, it's always important to know that, okay? But you'll hear me refer to them as axial filaments, and that makes sense because it goes down along the axis of the particular spirochete, okay? So this is going to be anchored or enclosed in the space between the cell wall and the cytoplasmic membrane. I kind of like this illustration here. You can see how it goes around. And these kind of spiral around, okay? And they help with movement, okay? It's a corkscrew-shaped bacteria, and it kind of moves like you would expect it to. A corkscrew, kind of like a corkscrew moves. Those cockleburrs, sometimes they're great big ones, and then you have the little bitty tiny ones, but they stick to you and you cannot get them off. Or we find we get them in our horses' manes and tails, and oh my gosh, they are a nightmare when you have these great big things. They're sticky, they stick to something, and they don't want to let go. That is what fimbriae is. They're small, bristle-like fibers coming around the entire surface of bacterial cells, okay? And they allow tight adhesion between fimbriae and epithelial cells. So epithelial cells, remember, are our skin cells. So it helps them to cling to our skin cells. So think of one of those cockleburrs being attached to your skin, okay? And it makes them stick there. So it makes them harder to get rid of. So it can contribute then to the virulence, or how bad, I could say, a particular microbe is for us, is how pathogenic are they, okay? You can also have this pili here, and that allows for transfer, which is good for the bacteria, bad for us, because this is used in bacterial conjugation. And I did link you a video on bacterial conjugation, which you really should watch, because it explains the whole process of how this is transferred, okay? Because this transfers things like antibiotic resistance, all right? So that's why I say it's good for the bacteria, bad for us, okay? You particularly see this in gram-negative bacteria. We are going to get into what a gram-negative and gram-positive bacteria is, but that's how we classify bacteria into two main groups. Most of them fall into those two categories. There are also some types of pili, like the type 4, that can transfer genetic material. help with attachment to things as well, and they can also work or perform the function of motility or movement. Okay. We've recently discovered nanotubules or nanowires, and these are thin, long tubular extensions of the cytoplasmic membrane. And these are used to transfer things like amino acids or to obtain energy by shuttling electrons to an iron or from an iron-rich substance. Okay, so it's very helpful for them. So bacteria protect themselves with or from environmental conditions with things like an S layer or a glycocalyx. All right, we'll also look at endospores. The S layer is basically thousands of copies of a single protein kind of linked together, almost like chain mail. Okay, chain mail is what, if you're sword fighting, this is what the guys would wear under their armor. It would be the chain mail. Okay, so it's a heavy netting of metal material where nothing can get in. Okay, so we only find this though when bacteria are in hostile environments. So C. diff or Clostridium difficile, Bacillus anthracis both can do this. All right, they don't always do this, but only when they are in a hostile environment. So Clostridium difficile, C. diff, is a really nasty pathogen of your intestines, and it causes really bad diarrhea. It is very, very, it's passed along very, very well. You find it in areas like nursing homes, long-term care facilities, and all. And it's really just particularly difficult to get rid of. Bacillus anthracis is anthrax. All right. All right, so the glycocalyx, this is a coating of polysaccharides or glycoprotein. So really large sugars or sugar protein units is what that is. They also serve for protecting the bacteria, okay? And then you can have some variants of that. One would be the slime layer. The slime layer is like a glycocalyx that's kind of everywhere, and that will protect against what we call desiccation, which is water loss, or loss of nutrients. And then they can have a capsule, which is much more tightly bound or stuck to the bacteria. It's denser, it's thicker, and it produces a sticky, or gives a sticky character to the colonies when we plate them out on an agar. So you can actually see that it looks kind of rough or sticky looking. All right, so here again, you can just kind of see those layers if they're here. Here's the inside. This would be the cytoplasm. Then this is the cytoplasmic membrane, the peptidoglycan cell wall, that S layer, and then the glycocalyx. Another particularly difficult for us to deal with protective structure for bacteria are capsules. This is a good stain of an encapsulated bacterium. And so you can see here we have bacilli, okay, and it looks like it might be diplo, okay, it might be strep. It's a little hard to make out in the picture, but this white clearish halo here is where the capsule would be. Okay, and again, that's protective in nature, and it's bad for us because it makes it hard to treat and get rid of the bacteria. All right, so getting into more detail of the functions of the glycocalyx. Streptococcus pneumoniae, which is one particular cause of pneumonia, you'll find out there are lots, and Haemophilus influenzae, which doesn't cause the flu but causes meningitis. Also, Bacillus anthracis or anthrax all have capsules. Capsules, like I said, are particularly difficult for us to deal with. So they therefore have a greater pathogenicity, which means ability to make you sick and harder to treat, okay? And so many of the pathogenic bacteria have capsules to protect themselves. And these capsules can protect against what we call phagocytosis. If you've had me before, you know what phagocytosis is. Cyto means cell. Phago means basically cell eating. Okay, so this is the process where our white blood cells will eat or engulf a foreign molecule in an attempt to prevent infection. Okay, so if they're able to prevent phagocytosis, then they're able to get around our defense mechanisms, and they can evade our defenses and multiply at will, which is not good for us. And then biofilms, all right? And these ones I said were like little colonies that kind of lived and functioned together as one. This can be as simple simple as the white plaque that forms, dental plaque or dental caries that forms on your teeth, okay, to slimes. That's going to be some slime produced by streptococci in your oral cavity. But this also is going to be when you've heard of somebody that's had surgery and they've had to go in and they have an infection and they've had to go in and have it scraped out. That's because it's a biofilm. They like to adhere to moist surfaces and foreign substances like catheters, IUDs, implanted devices, like if somebody gets a hip replacement and they can get a biofilm on the actual metal that they put in the body. And so that is very difficult and it's very hard to remove them because it's protective in nature. It's like you have all these layers and you can't get down to all the bacteria to treat it and kill it. So they have to go in and just scrape it out and just remove it. So this is just a good illustration here of how these biofilm can form. You get a few bacteria here. That's the first colonists adhering to the surface, and then they're going to divide and make kind of like a little mat, and then eventually they're going to create this complex community. And so you can see way down here, you can't get to these bacteria. You can't treat them.